Hi and welcome to Brown Biology. Today we will be talking about mitosis and meiosis in animal cells. Mitosis and meiosis are types of cell division, but they are different processes that result in different things. First, let's talk about mitosis. Mitosis is a diploid cell dividing into two identical daughter cells by IPP mat. IPP mat stands for the different steps that occur in mitosis. First, interphase has three parts to it: the G1 phase, S phase, and G2 phase. The G1 phase is when the cell grows. After that is the S phase, which is synthesis. This is when the cell replicates its DNA. Last is the G2 phase, when the cell grows again and continues with its normal process. After interphase, we have prophase, which is when the DNA condenses to chromosomes and the nucleolus disappears. Next is prometaphase, which is when proteins attach to the centrioles and form connectochores. Then spindle fibers align the chromosomes in the middle of the cell during a phase called metaphase. A good way to memorize this step is that the M in metaphase can remind you of middle. Next is anaphase, which is when the spindle fibers pull the chromosomes apart at the connectochores. Last, we have telophase. Telophase is when the chromatids reach the opposite end of the cell and membranes form around the daughter nuclei. Now the very last step is cytokinesis. Cytokinesis is when a ring of protein composed of actin pinches in at the middle of the cell, creating two new but identical cells. Now it's time to talk about meiosis. Mitosis and meiosis can be very confusing because the steps are similar, but it is extremely important to know the differences between these processes. First of all, we need to understand that meiosis is split into two phases called meiosis 1 and meiosis 2. Let's start with meiosis 1. After interphase, which consists of mostly cell growth, the DNA condenses into chromosomes and becomes visible at tetrads, which are non-sister chromosomes in prophase 1. What also happens during this time is the process of crossing over. Crossing over is the exchange of the genetic material between the tetrads. Spindle fibers then line the homologous pairs up in the middle of the cell during metaphase 1. The homologous chromosomes are then pulled to opposite sides of the cell. Then, in telophase 1, the chromosomes are gathered into separate nuclei and the cell divides. But the process isn't over. Now the two new cells must go through meiosis 2. In prophase 2, the chromosomes condense again, but the DNA is not replicated this time around, and crossing over does not occur again. The paired chromatids line up in the middle of the cell during metaphase 2. Then in anaphase 2, the chromatids separate and are pulled to opposite sides of the cell. In telophase 2, the chromosomes gather into separate nuclei, and the cells divide once again. Meiosis is then complete. Because the DNA didn't replicate during prophase 2, the products of cell mitosis are haploid cells. Cell meiosis are haploid cells. Also, these cells are different than the original cell because crossing over in prophase 1 created genetic variation. So that is meiosis and mitosis for you. Real quick, let's discuss some of the differences of these two processes. First of all, meiosis results in 4 haploid cells that are genetically different from that of the original cell, whereas mitosis results in 2 diploid cells that are identical to that of the original cell. There is only crossing over in meiosis, not mitosis. The pairing of homologous chromosomes only occur in meiosis, not mitosis. In mitosis, the cell only divides once, whereas in meiosis, the cell divides twice. Lastly, the process of mitosis creates any cell except for gametes, whereas meiosis creates gametes like sperm in males and eggs in females. This is why the cells produced in meiosis only have half the number of chromosomes. Then, when a sperm and egg meet, they then have the correct number of chromosomes, which is 46. Meiosis is for sexual reproduction, while mitosis is mainly for growth and cell duplication. Now that we have explained cell mitosis and meiosis to you, I'm going to show you a song about mitosis that might help you remember the process better. This song is written by Mr. W, but it will be sung by our own Christine. Thank you for watching Brown Biology, and I hope that was helpful.
Alright, everybody ready? Alright, let's go.
Interphase is subdivisions on the spot. Nuclear membranes intact as it starts. The cell's growing, cytoplasm flowing. Chromosomes are duplicated, DNA gets replicated. Chromosomes are spread out so they can't be seen distinctly. Below the nucleolus, the ribosome, bacteria, the nucleus are two centrosomes. They later make a spindle which are pulled by the chromosomes properties. The chromosomes condense. Each way to sister chromatosis like their necks. Each sister is a clone, the closest of kin, and essentially connects them like Siamese twins. The nucleus disappears and melts away. As the cell takes a ribosome, it is holiday. The centrosomes separate cells for formation, for separating chromosomes and cell elongation. Mitosis, chromosomal divide, report meta-anatophase divide, carrions go from one cell to two. Mitosis, how cells unite. In like prophase, metaphase, the nuclear membrane disintegrates. The centrosomes migrate to the cell's opposing sides, and between them the fibers of the spindle winding line. The spindle is made of microtubule cells which attach to chromosomes like kinetic cause a protein patch. That cells like the handle that the fibers can grasp. When they pull apart the chromosomes, splitting them in half. The spindle moves the chromosome with a so band to linear formation on the 50-yard line. With the equatorial dividing metaphase, the chromosomes are lined up on that letter place. Chromosomal improvement and a telophase divide. Carriers go from one cell to mitosis. How cells renew. Stem fibers growing in the kinetic cores. A cellular molecular mitotic tug of war. The sexually snaps. Since you get separated, now these chromatids are chromosomes. They've been upgraded. The snap of separation defines anaphase. The A part is spindled in different ways. Kinetic cores spin the fibers separate the dissociating waving good flag calling I'm gonna miss ya. Through the fibers pushing gobble like bones. Just a little watermelon. In telophase, membranes form around the chromosomes with strip of nuclei come on home. Mitosis, chromosomal improvement, anaphase divide. Carriers go from one cell to mitosis. How cells renew. Animal cells live in a microfilaments that form at the equator and they crush themselves in. Tear to the cells into pieces yet animals. That's cytokinesis, but it's different in plants. In themselves divides by building a new cell wall from the inside is gradually since vesicles of cellular glue complete then a wall divides cell in two. And instead of one mother, so we now have daughters too. But they're twins, kind of old but kind of new. In the sense of this is how you grew. And for a single cell, carriers reproduct the two. Mitosis, chromosomal improvement and a telophase divide. Carriers go from one cell to mitosis. How cells renew. Mitosis, chromosomal improvement and telophase divide. Carriers go from one cell to mitosis. How cells renew. Peace out.